There's a saying in medicine: when you hear hoofbeats, think horses, not zebras. Meaning when you're diagnosing, think of common causes first. But of course, those hoofbeats could be a zebra, even if it's unlikely. That's why the zebra is the symbol for rare diseases. And some are very rare indeed, affecting only a few people. But the overall number is considerable. It's estimated that more than 400 million people around the world have a rare disease. Historically, finding funding and treatments has been tough, but new DNA-based gene therapies are offering hope. Could they be a game changer? Then, 10 years ago, a landmark study totally upended what we thought we knew about preventing peanut allergies. It forced a complete reversal of guidelines at the time. And a decade later, we are now seeing the results. This is The Rundown. DNA-based gene therapies have the potential to revolutionize our understandings of rare diseases and offer new treatments. To learn more, I talked to University of Toronto researcher Rachel Harding about medical breakthroughs in Huntington's disease. Then, I spoke with Terry Piravalakis about his young son, Michael, one of an estimated 80 children around the world with a condition known as spastic paraplegia type 50. Terry shares the story of how Michael's life was changed, thanks to the first single-patient gene therapy trial at Sick Kids Hospital in Toronto. Rachel, Terry, welcome to The Rundown. How are you doing?
Great. Thank you for having us.
Yeah, thank you for having us. This is awesome.
Yeah, no worries. Rachel, let's start with you. Walk us through the recent Huntington's disease gene therapy. Specifically, what does 75% slower disease progression actually mean?
Yeah, so maybe let's start with what Huntington's disease is. So this is a rare, inherited neurodegenerative disease characterized by progressive, which means symptoms that get worse and worse over time, which are motor, so movement, thinking, and psychiatric symptoms as well. And everyone who has Huntington's disease makes a slightly different version of a protein called the Huntington protein. Okay. And so the gene therapy that was designed by this company, uniQure, is delivered by brain surgery, and it's genetic instructions that are packaged into this harmless virus that spreads through the brain and tells the brain to make less of this kind of harmful form of the Huntington protein. And so there's been a whole bunch of folks who've been in this clinical trial for quite a long time now. And we just got some data a few months ago now, which seems to suggest that there was slowing of symptom progression by about 75%. And so this is equated to symptoms that would normally progress in their aggressiveness in the course of a year. That would now take 4 years. Okay. So this is really exciting. This is the first time any company has shared any data that seems to suggest that they're able to slow down the course of the disease. We do have to be a little bit careful, though, because this is a very small trial.
So say how many people?
12 people.
Okay.
So, which is because we don't want to give brain surgery gene therapies to hundreds of people at once until we're really sure, are they safe? How well might they be working? We need to get some good signals from those first. And so we've just got 12 people. And the other problem with these kind of trials is it's not always practical or ethical to do a placebo arm. So actually what they're doing is they're looking at what they call a natural history cohort. This is where they track data from people who have Huntington's disease, but they're not in the trial. And we're trying to see how they would progress without the drug and then making that comparison. And so there's a bit of a debate about how good that is as a comparator, but nonetheless, it's still an encouraging step forward. And I always like to think about these super brave people who took this gene therapy, had this brain surgery before they knew there was any possibility this might be definitely okay. And they've been walking around for the last few years with this gene therapy in their brain, lowering the amount of this toxic protein, and how amazing that is.
Rachel, what would you say was the key breakthrough that allowed sort of these incredible results that we're seeing?
Yeah, I always bring it back to the patient community. The Huntington's community themselves are one of the most hypervigilant and organized communities, most well read up on everything to do with their disease, how to organize and how to best be involved with clinical trials. So their engagement with natural history studies, all those, I think it's over 10,000 people who signed up for every year. They go and they get certain measurements made. And if these people hadn't done that, we wouldn't have been able to do this trial and to make this comparison comparison. I think that's a really amazing thing. And it's not just the folks who have Huntington's disease, it's all their carers, their partners who drive them to these appointments with their clinical neurologist. That is what has really enabled things for this community, in my opinion.
Terry, you know all about this type of struggle. Your son doesn't have Huntington's disease, but can you tell us a little bit about your son's journey?
Yeah, Michael has SPG 50, or spastic paraplegia type 50. We were told, go home and love him. He'll be paralyzed from the waist down by the age of 10, quadriplegic by the age of 20. He'll never walk, he'll never talk. We went home and lost our minds that evening. I locked myself in a room. 24 hours later, we realized that we could do something, that gene therapies and ASOs or antisense oligonucleotides, there was therapies that could be done. And a month and a half later, I flew around the world. I met all the experts in gene therapy. We hired a team out of Dallas, Texas, and we started doing this therapy for Michael. This they call it a proof of concept. Prove that you can create a therapy. About a year later, we showed that it would work. It was working in mice, and it was working in cells, and we had to raise $4.5 million at that point. Our community in Toronto came together. We raised $4.5 million over 4 years, and we were able to safety test the drug, make the therapy, and treat Michael in March of 2022.
We have some photos of Michael that I want our viewers to see. How is Michael doing today? When was this photo taken?
This photo was taken well before his therapy. He had a major seizure that lasted about 3 hours.
Wow.
And they didn't think he was going to survive, and this is him waking up from that crazy seizure.
And then we have some photos here as well.
Yeah, that was when Michael was 12 months of age when they diagnosed him with this disorder. Well, they didn't exactly know what it was, but they diagnosed him with microcephaly. We went to Greece for a pilgrimage to try to pray away the disease, and yeah, this was just this is him. It's a happy little kid.
How is he today? Is this?
Yeah, he's good. You know, Michael was the most severe child in the disorder, but he's able to communicate with us, tell us what he wants. He hugs and kisses us. He shows affection. He's able to stand. You know, he's improving, but he was the most severe. There's a spectrum of disorder, right?
Right.
And so he probably will never walk. We hope that he'll talk someday, but if he's able to tell us that he's in pain, or he wants his diaper changed, or if he wants to go to bed, then when we're gone from this earth, we'll have a one. Yeah.
Rachel, what do these results tell us about the current state of disease-fighting medical research?
I think we're in a really exciting phase of drug discovery and finding treatments for diseases which have often been overlooked because they're not seen as commercially viable by really large pharmaceutical companies. Now we have technologies that allow us to almost copy-paste for different types of disease and to create therapeutics, hopefully, for many, many people. That's such an exciting place to be in our society.
Now, there's a lot of excitement in gene editing tools. We've heard technologies like CRISPR, CRISPR, but in terms of this Huntington's treatment, it did not involve that. Tell us a little bit about the technology. You touched on it a little bit, but tell us a little bit about the technology. Yeah,
so this therapy is delivered, like many other gene therapies, through this harmless virus. The drug itself is called AMT-130. The virus packages the instructions for something called a microRNA. This sticks onto the Huntington message molecule. This is kind of the in-between of our DNA genetic instructions and the proteins that they encode. And we have these message molecules that kind of are the in-between of those so that we can faithfully copy the DNA and turn it into protein. And so this microRNA sticks on to that message and then stops the protein being made. And so that's the technology. And so you can imagine that now we know how to do this for different diseases. We might find another disease where we want to switch off another gene or turn down the levels of another protein.
Terry, even when the science is promising for diseases like this, there are obviously some real barriers. You mentioned a number there, 4 million. That's just one side of the conversation there. But tell us a little bit about the barriers that that families are facing when it comes to these diagnoses.
Yeah, I mean, we're not meant to create therapies. Right. A family is not meant to raise $4.5 million, do toxicology in animals, find a manufacturer. This is not something we should be doing. And that's the barrier that we have, is that because we're not commercially viable, that companies are not going to spend $1 billion to make a therapy like this. There's no interest, and that's why when I had to quit my job and start a pharmaceutical company, or about
with no medical background, correct?
No medical background at all. Well, I had smart people around me, so it was a team effort. It wasn't just me; it was hundreds of individuals, and now I have a team of people that are much smarter than I am to get these programs to the clinic, show that they work, and then move them towards approval. Because the reality is, if we treat 8 children, we've really done a disservice for the community, because there'll be children born every day, there'll be hundreds over the next 20 years, there'll be thousands of children, and they could have been treated with this therapy, and they will not have unless we get it approved.
Can you help us understand how rare these diseases are? Like Michael's condition, how many people in the world have?
So Michael's one of 130 in the world. He's the only child in Canada. If you put in perspective, there's 10,000 rare diseases. 5% of them have a therapy of any sort, and 400 million people in the world have a rare disease. 1 in 10 of us will be affected. So someone that you love, will have a rare disease. Parkinson's, ALS, Alzheimer's, sickle cell anemia, hemophilia. Someone has a rare disease or will have a rare disease.
When you were starting your family, I imagine you didn't think that this was the fight that you were going to be fighting, that you were going to be an advocate for treating rare diseases. What other changes are needed to be successful in this fight?
From an individual perspective, when you're in this?
Yeah.
Well, I was an introvert. It doesn't show it anymore. But once you do 100 interviews, kind of breaks you afterwards. But you have to have a strong will. When we first put out our story, people were telling us to put down Michael, that he doesn't deserve to live, and you have to have a strong backbone. You have to have a strong will. You have to have a never give up attitude, and you have to have a support community around you. And luckily, I have an amazing wife and amazing children that supported us, an amazing community that came on and said, we are not going to let Michael have this disorder. We're going to be here for you. And then we were fortunate enough to meet amazing people along the journey to help us do what we needed to do to get there. It was, again, it's not meant for families to do this.
I have to imagine that you get emails, phone calls, messages from families who were in a similar situation than you were.
I get 15 calls a week. I had 2 just on the way here. It's nonstop because families are trying to save their children. They see my story and they want to do what I did. And some of them are successful, some of them are not. And it's something that we have to figure out. We have to figure out a path forward. Thankfully, technology is changing rapidly. And what we thought was non-curable 10 years ago is curable now. And if our children were born 5 years from now, it would be a different story. We'd have probably therapies right off the bat. Some things we need to do are newborn screening. We need to have genetic screening for every single child that's born. That way, when a drug is in the clinic, we can treat a 5-month-old, a 2-week-old, a one-month-old, and those children will be more towards a cure than a treatment. Because as you can imagine, it's like a sliding scale. If we catch them early enough, it'll be more towards a cure. If we treat them at 17, it's less and less of a cure, less and less of a treatment even.
Right.
So that's where we have to really step up, and governments have to start really investing in this new technologies and new innovations.
Rachel, Terry had mentioned a path forward. So I am curious, how can the lessons learned in the Huntington's case apply to the way we treat genetic diseases in general?
Yeah, well, I think doing any of exactly the work that Terry discusses, like this is hard and it's quite difficult. We need the community to mobilize both scientists and government to allow us to find new paths for how can we get drugs from ideas that scientists have in the lab to things that we can test and develop in the clinic into the most important part of this is getting the drugs to the patients. And one of the biggest barriers that we can kind of foresee with gene therapies in particular is the barriers for how you actually run the clinical trials, how you actually get approval, and then how expensive those drugs are at the end. Typically, we're talking in the order of maybe $1 to $5 million per patient, and how Health Canada and other approval agencies are going to oversee, administer that, and ensure that people can actually be treated with drugs that are maybe already available, but they're just so expensive. So that's going to be a problem we've got to figure out.
What kind of diseases might be in line next?
Yeah, it's a great question. I think I really like that Terry brought up this statistic that 1 in 10 people in the world have a rare disease. And so these are all kinds of different things. Some of them we really don't understand. We don't really know if they have a genetic basis or if it's some other environmental factors or something else altogether. But I think at least those with a defined genetic basis, there's a very obvious path forward for how we can use some of these emerging technologies with gene silencing or CRISPR, other kinds of editing technologies to try and find viable therapeutics. And I think that's where things are looking really exciting in the next 5 to 10 years.
All right, my last question to both of you. Terry, I'll start with you. What's your one hope for the future of genetic medicine in the next, say, 5 to 10 years?
My hope is that the technology has evolved to a certain point where a child is born, we do a genetic screen within a day, and on day 2 they're getting treated. And I think we're not far from that. I think in the next 5 or 10 years, that we'll be eradicating disorders. And that's why I say Michael was just born a bit too early. You know, if he was born 5 years from now, he would have been cured. Unfortunately, he was born now, and we had to do what we can. But that's our hope, is that we eradicate these disorders so that our loved ones will never suffer from these diseases.
Rachel, you get the last word.
Okay, I think something that's really exciting with these technologies is the underlying science didn't even exist maybe 10, 15 years ago. And so it makes me so hopeful about what is currently in the lab, like what scientists at University of Toronto and other places around the world, what they're developing and what possibilities they offer for the future. I don't think we even know what technologies will exist that could enter the clinic in the next 5 to 10 years. And that's really exciting.
Very exciting times. Rachel, we're gonna leave it there. Thank you so much for your insights. Terry, thank you so much, and wish your family all the best.
Thank you so much. Really appreciate it.
Peanut allergies are common and can be deadly. For years, parents were told to avoid feeding them to their kids under 3. But in 2015, that all changed. Research found that introducing peanuts as one of a baby's first foods actually cut the risk of developing food allergies. A study published in the journal Pediatrics this October says this updated guidance has had a big impact. I asked Josh Koenig, assistant professor in the Department of Medicine at McMaster University, about what the research shows and what it means for people who suffer from food allergies. Josh, great to have you. How are you doing?
Doing very well, thanks. Thanks for having me on.
Of course. As someone who researches food allergies, how groundbreaking is it that we are seeing peanut allergies plummeting in children?
I think it's a very promising sign. As you know, the recommendation shifted to from basically avoiding allergens early on in life to early introduction and early feeding at about 4 to 6 months of age. And this happened around 2017, which was the first recommendation. And in 2021, there was a more expansive guideline. And, this is based on a large study called the LEAP trial, where they found that early introduction had a massive impact on becoming allergic to peanut very particularly. And I guess now is probably just around the time that we would be able to see the beginnings of that signal that, we really are impacting the incidence of this, of the disease. And, I won't say that the data are, there's like an 81% decrease in sensitization in the LEAP trial. That's not what we're seeing yet, but the difficulty is, that's a very controlled environment. And in the real world, when, you're in clinics, and there's clinics all across the country in the US, which is where this research was done, uptake by patients, these are all questions that are still open, but it's a really positive sign that we are, for the first time, I think, in modern history, seeing a decrease in the rate of peanut allergy.
In layman terms, can you help us break down the science here? Why does introducing peanuts and peanut products, like peanut butter, lead to a drop in allergies?
So for 98 to 99% of us, peanut isn't a problem. It's only in a much smaller fraction, 1 to 2%, where people become allergic. What typically happens when we eat a food is we become what we call tolerant to it. Our immune system works in tandem with our intestine to say, those foods are okay. We need to take those things in to get nutrients from them. So we don't want to have a bad reaction. But in 1 to 2% of people, that doesn't happen. Instead, they mount an immune response against the allergen. And then that immune response can have, the effects of an allergic reaction when they see the allergen again. So what we think is going on is that by providing the allergen early on at that 4 to 6 month time point, you're introducing the allergen in a controlled way, in a controlled environment, such that the immune system hasn't had time to accidentally be exposed to the allergen in a negative way that would make you allergic. So essentially, we're just taking advantage of what we know about how the immune system works and using that to tolerize people before they have the opportunity to become allergic.
I can imagine there are people who are looking at this study and are quite excited. They may not be allergic to peanut products, but they might be allergic to something else. And I am curious, is it recommended that we try other allergens like tree nuts or eggs?
So I'm not a clinician, so I can't provide clinical recommendations, but I can say that, in speaking with my clinical colleagues, there are actually schedules that are recommended for early introduction of essentially all foods because they think that that really is the way that we are going to, decrease the incidence of new peanut allergies. But as clearly evident by the study, it wasn't an 81% decrease that we saw, in these patients. It was much less than that. And people are still becoming allergic. So I think that, yeah, following those schedules is a really important piece of this. But another important thing is that people are still allergic and are becoming allergic. And we do need to find ways to deal with those people as well.
So I want to clarify. So people can still be allergic to peanut products or other products, even if they've had them at a young age.
So, that's a very good question. I suppose we don't know exactly the answer to that question. What tends to happen is that people on that first introduction to the food, their child have a reaction right away, and then they end up having to go to hospital, and then they end up being diagnosed. So essentially, the thought process there is that they've already become allergic before the early introduction.
Peanut allergies has been getting a lot of headlines, not only from this study, and you had mentioned the US. I actually want to pull a clip. This is from Robert F. Kennedy Jr., who is, as we know, the US Secretary of Health and Human Services. He gave an interview recently talking about peanut allergies and exposing infants to peanut products. Let's have a listen to that.
To me, that is not a convincing hypothesis because, number one, in my own personal experience was my house was so filled with peanut butter. And I was eating peanut butter 2 meals a day. And my wife, when she was pregnant, was eating peanut butter. And the kids were given peanut butter from as soon as they could chew food. And yeah, we still had peanut allergies in our house. We need to look at aluminum adjuvants and vaccines, which fit the timeline perfectly. The increase of aluminum is in lockstep, which began in 1989, which is in lockstep with the expansion of peanut allergies.
As a researcher, I have to ask, what is your response listening to that video?
Um, well, on the one hand, anecdotal. It's really unfortunate that Mr. Kennedy's children have peanut allergies. But this study involved tens of thousands of people, so this is a much larger aggregate than an anecdotal study. Um, you know, eh, there really isn't any evidence that would support the notion that alum or aluminium in uh, vaccines is associated with allergy at all. In fact, there's been multiple what we call systematic reviews, which is essentially looking at many different studies that have tried to look at this, and essentially there's no connection between vaccination and becoming allergic as far as we can see. Um, and I suppose I have a small issue with the timeline there because I'm pretty sure that alum was first introduced in like the 1930s and has been in vaccines for the better part of 70 years or something like that. And, the prevalence increases that he's talking about generally are from the 1990s onward. So I'm not really, I'm not sure I buy that as an explanation. We really don't know why the prevalence of allergy has been on the rise. And I think that that's a major issue. And I don't think, in contrast to what Mr. Kennedy is saying there, I think, there are many fantastic researchers that are trying to figure this out. They're trying to figure out what was been causing the increase in these allergic diseases. And, really consensus hasn't come about on that. But I don't see any evidence that it would be anything related to vaccination. In fact, the opposite. I would say studies are kind of suggesting that, for example, if you get a vaccine, you're less likely to have eczema.
Hmm. Well, I want to pick up on that in terms of the research that's being done. Are we any closer to identifying the what, like what causes food allergies?
Well, unfortunately, not really. Again, not for a lack of trying. The difficulty with this is people become allergic very, very early. So think about how we could know this piece of information. Most times we know that someone's allergic when they've eaten an allergen, had a bad reaction, and gone to hospital. So trying to figure out what happened before that is difficult because we don't know of out of 100 people, maybe one or two might become allergic to peanut, right? And it's been very difficult for us to pin those down. And the things that we have found that have made some associations, for example, like peanut oils in baby oil, those things resulted in changes. So you won't find peanut oil in baby oil anymore because of the possibility that you might be sensitized. And researchers found that out, and we enacted changes to, avoid that potential issue. So I think these large epidemiological studies are likely to reveal new things in the future. But for right now, I think the bigger issue is how can we engage in prevention, and then how can we engage when people are allergic to basically stop them from being allergic and give them the opportunity to live a regular life.
All right, I want to change gears a little bit. I want to talk about we're in an age of disinformation and misinformation, particularly when we talk about healthcare. And I want to talk about a Facebook post that claimed there was a vaccine for peanut allergies. And
Right.
I have to imagine that you and your colleagues who are deep in this research in allergies would probably be one of the first people to know that this was in the works. And so this was not the case, of course. You and several of your colleagues at McMaster put out a statement to say it wasn't true. But people are curious, is there a vaccine in the works for peanut allergies?
So yeah, I mean, I think that was an unfortunate situation of AI hallucinating based on some potentially truthful information, and then a lot of essentially gibberish. We are engaged in multiple studies testing potential therapeutics in food allergy, and trying to figure out what options are available and what's going to be effective. Early in development, we do have some studies looking at what would essentially be vaccines. But, they're nowhere near the point where we're giving them to humans and seeing an impact in the human. And, it's very unfortunate because I would absolutely love to say to everyone here, I have the solution. I don't, but I can tell you, I don't sleep very much because I'm on the case and I'm always trying, I'm always working towards it. And so are all of my colleagues. So we're working really hard towards this. And, we have multiple lines of inquiry to try and do this, as well as trying to figure out, what causes allergy, what makes allergy persistent in people. And we're hoping through those research programs, we'll be able to find good interventions that will have an impact on people.
Well, let's stick on that point about sort of battling the misinformation, disinformation. As a researcher, how is that battle? What do you do?
It's a really fantastic question. And, I think engagements like these are good opportunities. I definitely know that my colleagues and I were, overwhelmed by the amount of work that we have to do, both within the research program and then beyond. So, the notion of doing engagement is we don't do it as much as we would like to. So I think one piece that we need to do is engage more with the community. And when I engage with community members, which we do quite a bit through Food Allergy Canada, which is a partner organization, a fantastic partner organization, I find that people tend to go, oh, okay, well, now here's a face to a name. But the difficulty is that the disinformation process is so fast, and it's really hard for us to, cut off everything that comes out and make statements about everything. When the statements are, very clearly false and very clearly about us, it's a little easier, like in this disinformation campaign or this disinformation AI post situation. But, I think that this is an open question. How do we as researchers demonstrate what we're doing to try and impact this problem and, get the trust of the general community back?
Josh, really appreciate it. Thank you so much for your insights, and your research is really exciting stuff. Thank you so much.
Thanks so much for having me.
Today is Giving Tuesday, and we're asking you to help us support media literacy and keeping Ontarians informed. Donate before midnight tonight at tvo.org/givingtuesday, and every gift will be matched up to $250,000. Donate $100 or more, and you'll get your very own limited edition retro TVO toque. Your support builds a stronger, more informed Ontario. Until then, we'll see you tomorrow.